So I want to start with this graph here. I made this graph by scraping some data from PubMed's advanced search. I searched for some keywords that contained the single-cell RNA-seq string and similar one. And what we can see is that even by looking at a single database, we can observe how the growth of popularity of tools like single-cell RNA-seq in the past 10 years has been growing at a steady pace. And I wanted also to show this other graph, which shows the cumulative number of tools, again, in single-cell RNA-seq that we see available. Half of the tools that you see here are in R, the other half are approximately in Python. And for looking at, for counting these tools, I look at the single-cell RNAtool.org website that is a lower bound, let's say, of a number of tools. And again, we can see that the cumulative number of tools is growing at a steady pace over the years. And with the growth of number of tools, along with increasing capabilities, unfortunately, there are also challenges that can arise. And this is highlighted in several study reviews. Here I put one, for instance, that shows that discusses how significant research time is lost, usually by PhD students with no formal training in software development, in re-implementing already existing software tools from often inadequate literature descriptions. And the problem is, as this sentence highlights, not just about the time lost in redundant work, but also reproducibility challenges, because when re-implementing the description or the code is not always easy to reproduce. So I want to focus on the second graph that I show, on the number of tools that is increasing over time, and zoom in a bit more on the history of single-cell RNA-seq tools in Python. So around 2018, 2019, ScamPy and data have been introduced for storing single-cell expression data and performing analysis procedures. In a few years from that, tools for probabilistic deep learning on single cell, like SCVI tools, have been developed, and this has been followed by other tools for specialized analysis up to single cell multi-omics methods, such as Muon. And it was around this time that scientists working with single cell data quickly realized, the developer themselves, that working with these different tools was posing interoperability challenges. You should be able to install ScamPy, but also SCVI tools in the same environment, and not always was easy to exchange information between these tools, even if they were installed at the same time. It was around this time that the developers of these tools gathered to form scverse. Scverse is a consortium of open source tools that tries to address the interoperability challenges and data overhead challenges that I show in the last slide. So I'm a core developer of scverse, and I wanted to discuss this problem because the reason why we created scverse is that we wanted to accelerate science via interoperable foundational software. So the way that scverse operates, the way we try to address these challenges, is by having a team that spans multiple expertise and that unites the developers and maintainers of the tools that I showed in the previous slide. So scverse starts with core data structures like ndata and mudata. And on top of the core data structures, scverse developed and maintain a set of core tools such as ScamPy, Muon, SCVI tools, and SquidPy. And this forms a base, a robust software base for a larger ecosystem, which is developed by the community. And the ecosystem covers a series of use cases for single-cell, multi-omics, and also spatial omics, as we will see later in the presentation. What we do is that we support the community by promoting best coding standard, and we do this by online training, but also via in-person hackathons in which we explore ideas together and we show the best practice. So this graph here shows a snapshot of the monthly download in PyPy for various single-cell methods of scverse. And we can see that currently we see a steady growth and adoption. And now I want to focus in the bottom part of the slide, the missing one. And to do this, I want to talk about spatial. Quickly check that the chat is not showing messages. Okay, like regarding audio. Yes, I want to talk about spatial. So we define spatial technologies as experimental technique that allows to detect molecules quantitatively along with positional information. An example of a non-spatial technology is single-cell RNA-seq, because despite being quantitative, doesn't come with positional information after the tissue is dissociated. An example of technique with spatial information is in situ hybridization, followed by microscopy. In this case, we don't have quantity information that allows to compare different areas of the tissue reliably or different samples. And to overcome this, there are spatial omics techniques. Here is an example in which, for the Drosophila embryo, we can see rich position information along with quantitative readouts. And I want later in this talk to focus on a particular technology, Visium. This technology comes with two types of information: a high-resolution tissue image, a microscopy image of H&E stain slide, and then gene expression information for the space in this tissue that comes as an array of expression with these hexagonal patterns of capsule locations. And here I show two genes that shows expression in space. So here is a zoom of the same plot. We can see that the capsule locations, so we can read that they are 55 micrometers in diameter, so the information is not at a single cell level. Each of these capsule location contains multiple cells. So in the context of spatial omics, now that I introduce what we mean with them, we see that the growth is even higher, is even faster if you look at the trend from PubMed like before, that for single cell data. And one of the reasons is that the landscape of spatial omics technologies is extremely heterogeneous. There are multiple types of technologies from academia or from commercial solutions. And not only there are different types of technologies, but also within the same technology, for instance, Visium that we just saw, or Xenium, there are multiple types of versions. Each of these versions is defined by a unique combination of file formats for storing the various components of spatial omics, such as raster data, geometrical data, or annotations. And which is already quite prevalent, is made even more complex, is made even worse by the fact that we see the emergence of spatial multi-omics readouts in which consecutive slices or the same slice is analyzed by different spatial omics technologies that come so with different file formats. And regarding the number of spatial omics technologies, here is a more in-depth of the graph below. So we can see that the number of genes per dataset over the year is increasing exponentially. And we can see that we have an exponential growth also for the total number of cells per study. So not only the complexity of the file format, but also scalability becomes a pressing challenge as the dataset grow. And the data also grow in a different sense than this above, because we can see that even if we consider images, with a single data set, we may have already a large image that is difficult to process, but we see also multiple overlapping slides present in the same study. So not only the number of cells for each of these tissue slice increases, but also the number of such slices can increase, and they can lead to complex data sets that require spatial alignment and the handling of a large number of large images. And there is another challenge, the challenge of interoperable analysis. So let's start with an example that considers Visium raw data. So When you have a Visium data, you can start by visualizing the data set, or you may want to consider the image part of the data set. We saw that Visium has an image and some gene expression. So you may want, for instance, to apply an image processing algorithm to the image part, or maybe you want to perform some analysis on the gene expression. And when you do this, you want to save the results back to disk. But this is when the complexity of the formatting increases, because when you save the analysis result, you go increasing the number of entries that are present in the raw data. Maybe you save some language-specific components, such as some Python-specific component, and this creates problem in downstream tasks. Like visualization becomes more complex, and if you want to analyze with a different tool this new set of data, maybe a tool from a different programming language, Jot is in the R programming language, not in the Python programming language like SquidPy, then you need to perform time-consuming data conversions. Even when these data conversions are performed, saving the new results from this new tool may increase even more the complexity of the file format, maybe adding a new programming language on the equation, and this hinders reproducibility. So we see that this is just a toy example of some very simple data with 2 steps. Real pipelines can have 10, 20 steps, and this shows how the lack of interoperability between file formats and analysis methods can really hinder reproducibility. And the last motivation part of this talk is that we are currently witnessing a scientific revolution that is fueled by artificial intelligence and the availability of a large curated data set. For instance, the latest Nobel Prize in chemistry was awarded to 2 computational scientists, Demis and John, for the prediction of the folding of proteins. And the reason, one of the reasons why it was possible to build such a resource on top of computational innovations and the skill of the scientists before, is that the data set that was used to train the model was extremely rich and curated. And I'm talking about the Protein Data Bank, which is an effort that started in 1973, when it was the dataset was composed by only these sequences, these 9 sequences, the proteins here. And over the years, there was a growth. In this case, is a quadratic growth, so quite prominent in the number of entries available in the database. And with spatial omics, this growth is even faster. So probably we will not have to wait for several decades to have breakthroughs in artificial intelligence for spatial omics. But we believe that by having reusable data structures that simplifies the coding time and make data storage more harmonized, we can really have the fuel for new machine learning methods. And so all the reasons that I described above are the motivation that the reason why within scverse we decided to work on a foundational infrastructure to accelerate spatial biology. And in doing this, we also decided to join forces with other players, in particular the open microscopy environment, some developers from OME, Open Microscopy Environments, that has experience in dealing with large images and standard formats, and some developers from Napari. Napari is a tool for interactive visualization in Python. And together we decided to address these challenges by building the SpatialData framework, infrastructure for data storage, manipulation, and visualization of spatial omics data. And also I want to highlight it from the beginning, it's not an analysis library, rather it's an infrastructure for other analysis libraries, such as SquidPy, to build on top and generalize their approach. So here I wanted to show the philosophy and the components of a SpatialData framework. We started by introducing a modular representation for spatial omics. We realized that each spatial omics dataset can be abstracted as a combination of simpler elements, such as raster geometries or vector geometries, and these pair with annotations that can enrich the type of information that is present for each of these elements or spatial layers. What we allow is that the user can stack together these various elements in any order, any number, even 0, and in doing this, the user can make use of a system of coordinate transformations so that the various layers or elements are aligned in space, and this allows, for instance, to represent a pixel space or maybe a physical space in micrometers and so on. Finally, when we write and read, we do it on disk by following an extension of the OME and ZFF specification, which is an open standard in bioimaging. And we write in ZAR and Parquet, which are two language-agnostic performant storage options. And I want to explain a bit more this part. So this is how writing in ZAR, following the OME and ZFF specification, looks like. Let's imagine that we have a large microscopy image. This can be, for instance, 50,000 by 50,000 pixels, so very large. What the format dictates is that the image will be first chunked, and instead of having a monolithic file, now we can save the various chunks separately and also access them separately, but also the image will be downscaled multiple times. Here I put a downscale factor of 500 and 1000 for graphical reasons, but usually it's like 2, 4, 8, 16, and so on. And each of these downscale images is also chunked, and this storage, which is called pyramidal storage, allows an access of the data in a similar way of Google Maps. If you want to see the details, you can zoom in, but when you zoom in, you load only a small chunk at high resolution. And so now that I discussed the file format, I want to discuss also some other tools that we developed on top of the framework. The first is called SpatialData IO. This is a Python package that allows the ingestion of spatial omics data from different standard technologies, for instance, Visium and Xenium that I introduced before. And once you have an in-memory spatial data representation, you can save to the ZAR format and also then read back. And when you read back, you gain the performance improvements of a file format, such as the one that I showed before, that allows the performant access of large images. We also develop, this is an example of SpatialData IO in practice. We can see 3 different technologies, all from the same company, 10x Genomics. These different technologies differ in resolution. For instance, on the center, we have a single molecule subcellular resolution and the number of genes detected. For instance, on the left, we have a transcriptome-wide readout, while in the middle, we have the readouts of up to 5,000 genes. So using SpatialData IO with a single function, you can read all this data on disk that otherwise will be highly heterogeneous and non-compatible. And when you have the object in memory, you can perform operations on it. And the object is describing a modular representation of the various technologies in the following way. All these technologies contain one image, or to be precise, a series of images. For Visium, as we saw, we have a combination of shape information, the circular capsule locations of 55 micrometers of diameter that are annotated by gene expression table. For the Xenium data, we have transcript locations encoded as points. We have also cell information annotated by gene expression, now represented not as a vector information, but as a raster information. And also we have this information for nuclei. On top of this, we also have an approximation that is available from the raw format of the same high-resolution segmentation masks into vector set of geometries. These are useful for visualization, for instance. And finally, on the right side, we have a dataset that is very similar to Visium, but now Visium HD has a different shape for storing the capsule locations. Now we have square bins, but also we have different resolutions, 2 micrometers, 8 micrometers, and 16 micrometers for the square bins. Each of these is annotated by a different gene expression table. So as we can see, the the type of geometries for each spatial omics dataset can be very different, but all of this is encoded in a unique spatial data object. And once you have the spatial data object, you can perform also some downstream operations. We start with data visualization. We developed a Python package called the spatial data plot that allows static composable visualization operations. For instance, here we can see that we build the plot step by step, visualizing the images first, then we visualize one scene on top of the capture geometries, and then we also set some parameters of the plot, such as the title. Here is a more complex example for the Xenium technology, in which we show again the image and gene expression, but now also we show for the same gene the transcripts, and we can see that the transcripts are accumulated more dense where the expression of a cell is higher. We also developed a Python plugin for Napari that allows interactive, not static, visualization of spatial omics dataset. And these are some screenshots of different technologies with spatial annotation that can be visualized with Napari. And to this topic, also I want to show this animation that portrays how we can visualize, we can zoom in very performantly, different align modalities, and we can also annotate some regions of interest that then will be saved in the storage format. So will be all these now areas, these shapes will be ready for downstream analysis in Python. Here I was screen sharing my Mac to my iPad. And I want to finish this description of spatial omics spatial data framework by also saying that we implemented a series of operations of a spatial data object. So the spatial data framework is not an analysis framework. Still, some APIs can be used for analysis of data manipulations. For instance, you can apply coordinate transformation. You can, for instance, as a use case, rotate an image with this. You can have queries to crop the data, so you can subset the data set on a particular region of interest, like the one that we annotated before with the iPad. We have also an interface with two PyTorch, so we can generate deep learning data set from spatial omics spatial data object. We also implemented spatial aggregation operations. This allows to align, to perform, to quantify metrics across modalities by combining a signal from a modality to some shapes from a different modality. And also implemented some functions to have an interchange between raster and vector representation for the same data. So let's see a bit more in detail these three operations here. Let's start with spatial queries. So assuming that we have a spatial omics dataset, in this case we have a Visium dataset, and let's assume that we annotated three different regions. What the APIs for spatial queries allow for is that you can subset the image, but everything that is aligned with an image, such as the segmentation, the circular capsule locations, in this case, to match this area. So you can query a portion of a spatial omics dataset without having to worry about coordinate transformations. And the syntax here is shown by this single call of a polygon query API. Here is an example of the aggregation operation in which you have multiple cells annotated by cell types in this example, and we want to count fractions of cell types within a region of interest. And this is the region of interest for the immune infiltration compartment of this duct in a breast cancer dataset. And this is the one we annotated before with the iPad. And the result is a fingerprint of the cell type content within this area. The operation to perform this is again a single function in Python called aggregate. And aggregation can be done in very general ways. We don't just aggregate cells, in this case where cells are represented as circles, annotated by some gene expression, aggregated into some polygons description. But there are much more general cases in which you aggregate points by a segmentation mask and so on. And all these cases, thanks to the use also of rasterize and vectorize, are supported by the spatial data framework. So you can focus on what you want to aggregate without having to worry of the specific data type that is used. So having showed this, I want to now show a snapshot. So this is the missing row from the plot below. We see that we currently witness a steady growth and usage of the framework, but we also I want also to point out that with growing adoption, there is even more the importance of maintenance and ongoing improvements. So now in this second part of the talk, I want to focus on what happened after the orange line, so what we developed after the paper was out, because the paper is, as of now, already 1 year old. So we want to start with different technologies that are now supported. We decided to structure the SpatialData I/O as a separate package than the main SpatialData package. And the reason is that we wanted to encourage the community contributions. So here I want to thank the community that really helped in supporting many new technologies. A subset is the one that you see in the slide here, and these are now available in the SpatialData I/O package. In particular, I want to focus on the Visium HD technology, which I already showed before is the high-resolution version of the Visium technology, the one with many square bins. So the square bins are arranged in I have this picture here. They are arranged in a sackboard-like grid, in a square grid, and they are very high, very small in size. So when you look at such disposition, such arrangement, you are encouraged to think of the data about being a raster data, not a collection of vector geometries, but as images, where each of these square is a pixel. So to do this, together with Can10, we developed the two polygons and rasterize APIs. Yeah, this I introduced even in the first part of the talk. And also we developed a function called rasterize bins that perform rasterization by exploiting the fact that we know that the data is stored as a series of square bins. There is a problem. The Visium HD data is so large, and also it's so compressed, it contains a large degree of sparsity that we cannot rasterize the full data set. If we consider Visium HD and a full rasterization, this will lead to 180 gigabyte as a dense image. And for technologies that are similar to Visium HD, such as StereoSeq, we will have even larger dataset after rasterization. We talk about more than 2 terabyte of dense image. So this is the reason why we developed on-demand approach to rasterize this type of data. And we did this by doing this. We developed this method by not having to create a new data type, but really interfacing with the existing data structure. So the user can reuse as it was the code that was available for images, but now the images are actually lazily computed for these bins when needed. And an example of this is that after developing the rasterize bins function, we could plug and play directly the Visium HD dataset in Napari. So here you see these are not accelerated, these animations that we scroll between different genes of interest in Napari SpatialData, and we saw a 1000x speed up in visualization of Visium HD data in Napari. So it's not typo here. But we realized also in doing this that if we were able to accelerate so much the visualization of vector geometries, there was probably a big room for improvement in visualizing vector geometries in Napari. So this is the reason why, with the funding that we received from CCI for the SpatialData project, CCI, the Chan Zuckerberg initiative, we funded developer time for addressing some of the performance challenges directly in Napari. So Gregor, here I put some of the screenshots from the various conversations that Gregor started, implemented a 25x speed up in general in Napari. So not just for spatial omics users, but in general Napari users. And this number is increasing every time I revisit this slide. So I'm really excited for this. Also, in the topic of performance visualization, together with Sonia and Clarence, we accelerated the speed of the SpatialData plot package by using, again, a rasterization approach with the data shader. And this has been very beneficial for the users. Also, in the last year, we saw that the community is enabling more and more interoperable analysis of spatial data objects via growing ecosystem. So being modular, the spatial data objects already allows to extract a single component, for instance, the table. The table is stored as an end data object, and reuse existing methods that work for end data objects, such as SCMPy. So for instance, you can perform quality control operations using SCMPy on an end data object from a spatial data object. But you can also consider the object as a whole and analyze it with external tools. Well, there is SquidPy that we developed as scverse, but also here is a list of tools from the community. Currently, we count 22 packages that use spatial data, and from the GitHub statistic, it is 98 repositories. And in particular, I want to highlight two tools. One is SOPA from Canten, which is a pipeline for segmentation, segmentation, quality control, and visualization for spatial omics data. And the second is Bento tools from Clarence that allows the study of subcellular compartments for single molecule spatial omics data. In particular, on the topic of interoperability, I want to dive a bit deeper in SOPA from Canten. So SOPA allows interoperable workflows between 10x Xenium Explorer, this visualizer tool from 10x, very popular for Xenium data, and the spatial data and scverse ecosystem. In fact, if you have Xenium data, SOPA allows you to save, starting from Xenium Explorer, aligned spatial data objects. You can now work within the scverse ecosystem, performing clustering or cell annotation. You can go back to Xenium Explorer for visualizing these new annotations, and now you can also select some regions of interest from the Xenium Explorer interface and go back to having a cropped spatial data object using the spatial query operations. So this is an example of interoperability between an open source Word and a proprietary tool from 10x, and this was allowed by having standard file formats both from our side and also from 10x Genomics side. They developed an open format that 10x Genomics is using for Xenium data. And on the topic of interoperability, so we saw an example of combining different tools, and this unlocks new capabilities and analysis. So this is what we can already do. We can start with Xenium raw data, convert this, parse this into a spatial data object, maybe save it to disk, and then visualize it interactively. There is also a link with the test, which is a visualizer written in JavaScript from the Gallenborg lab and Mark Keller. And we can also use the scverse ecosystem for Python-based analysis or a link to deep learning libraries, and then save a result in a spatial data object. What is not yet available, so this is really work in progress, so we'll say even like more highly work in progress, is combining Python with R for the analysis of spatial omics data. So what will be our goal, and the goal of Bioconductor, is to enable the user to reuse the large corpus of statistical analysis methods from Bioconductor or geospatial statistics methods, and perform analysis on spatial data object and then save a result again on spatial data object. So this goal will really unlock even more analysis capabilities. So to work on the goal, what we are doing is that we are teaming up with Bioconductor, and we met in a series of hackathons. The most recent was in November, a few months ago in Basel. This hackathon was sponsored again by CCI, that I want to thank. And what you see here is some work in progress from the Bioconductor team from the hackathon. This work is led by the Bioconductor team, in which, thanks to the language agnostic spatial data ZAR disk format, a spatial data object could be read and written to by Bioconductor tools. And in particular, we show an example of unlocking readers from Python now. So the readers are already implemented in Python, and thanks to this, following this path, the data can be accessed within R without having to code new readers. And also here is an example of visualization of spatial omics data with ggplot. You can see more on a preprint that we released after the hackathon. And on the topic of interoperability, I want also to give some hints on the next topic, on the next challenges we are working on. So the way we encode coordinate systems and coordinate transformations is by implementing, by a forced implementation of a part of the specification of NGFF called RFC5, the specific one. This is developed by John Bogovic at the Zanilia Institute, and is a way to represent coordinate transformations and coordinate system in a language agnostic way. So latest hackathon of NGFF, the NGFF community, we agreed that the implementation of the coordinate transformation that currently is available in the SpatialData package will better suit to live in a more general package. So I'm together with the other devs, we are moving the code in a more general package called OMSARS Model Pi, and this will make the code more maintainable in the long term, but also accessible not just to the spatial community, to the spatial omics community, but the geospatial and the bioimaging community more in general. So this is a large refactoring that will improve the interoperability between NGFF and SpatialData, and I'm really excited for this direction. Finally, I want to go toward the end of the talk by picking up again the topic of large data sets that can accelerate deep learning applications. So here I want to highlight that last month the CZI SelixZine census database expanded to SpatialData support. So census is a large, very large database of more than 110 million cells, single cells, that allows APIs for slicing and querying the data with high flexibility, and also exporters to various formats in various programming languages, including NData and recently also SpatialData. And SelixZine census is implemented using technologies such as TileDB and TileDB Soma. And in the latest release, now we introduce also SlideSeq and Visium datasets, so two particular technologies from the list of spatialomics technologies, and exporting this to the SpatialData format. So I'm really excited about this. And this is an example of having a large curated dataset that is ready for machine learning applications. On top of this, so this effort focuses on Visium and SlideSeq data. In parallel to this, within the team led by Tim, we are developing a public database of curated spatialomics dataset that will include Visium, SlideSeq, but also more general spatialomics technologies such as Visium XD, Xenium, Merscope. And the way we do this is that we start from the raw output of the analysis pipelines. We use SpatialData.io to parse this into a SpatialData ZAR format on disk, and also include some metadata that is provided by the technologies. And on this, the data will be uploaded to a data lake that is public, but also we're working together with Lamin AI to allow for a GDPR compliant infrastructure, and also having privately hosted data, so that this approach can be used also when the data can't be public. And on top of this database, there is a link to Vitesse, the browser-based tool for interactive visualization of spatialomics data. And also we have a function that allows to query data metadata directly from the cloud, similar to what I showed in the previous slide. And also there is a way to do local compute and upload the result, so that we can combine the cloud database with local training, for instance, training of a deep learning model. And this is still work in progress, but I am excited that next month, well, in 2 weeks, 3 weeks, we will present a preview of a SpatialData database at the hackathon that we are organizing in collaboration with Oken. So it's scverse hackathon that is hosted by Oken in Paris. We hope that also after the hackathon, when the work is finalized, we'll be able to release as soon as possible the database to the public. So with this, I want to show the last 2 slides. The hackathon that we had in Paris is one of a series of several hackathons that we organized over the years. We try to cover different countries. So we did this in Austria, Germany, two times in Germany, but also a conference in Germany last year. It was our first conference. And then in Switzerland, UK. And last year, for the first time, we expanded outside Europe in the United States. And in the future, so this is really unconfirmed. I put a question mark and I mean it, but we are trying to organize an event this fall in the Bay Area in the United States, in California, and also in spring 2026 in South Korea. So really excited about expanding also beyond Europe. And with this, I would like to conclude. Introduce the SpatialData framework, which is composed by a data standard for spatial omics. We implemented spatial operations in Python, for instance, aggregation, ROI, region of interest selection, and rasterization. And the current work is on the topic of interoperability to strengthen the bond with NGFF and different programming languages and cloud support. And with this, I want to thank all the people here, in particular Giovanni, Kevin, and Isaac are the other co-fourth authors of the SpatialData paper, and team and voter are part of the core team, and all the collaborators, our PIs, and company industry partners that helped with the tool. Finally, I want to thank our partners and institutions, and I want to thank you for your attention.